Up to this point, we've been studying DNA, protein synthesis, and genetics. In this chapter, we're going to tie all those things together so that we can see how they are all interrelated. This is chromosomes, mapping, and the meiosis inheritance connection, or sexual inheritance, chapter 13. We're going to begin our chapter by studying the scientists who contributed to our information of inheritance. We'll begin with Correns. The year is 1900. He was studying hawkweed plants in order to verify Mendel's work and to prove that his laws of segregation and independent assortment were correct. In 1902, Walter Sutton was studying grasshopper cells, and he discovered that chromosomes occurred in pairs and that during meiosis those chromosome pairs split, each going to their own cell. Prior to this, we had no knowledge of chromosome pairs or their movement during meiosis. In 1903, Sutton also continued his work by discovering that chromosomes were composed of genes. This led to what is known as the chromosomal theory of inheritance. This theory states that chromosomes are linear sequences of genes and that patterns of inheritance can be explained by assuming that genes are located at specific locations on the chromosomes. In other words, if you know where the genes are located and you know the patterns of those genes, then you can determine the patterns of inheritance. Correns returns to the stage in 1909 by publishing a paper about his work with 4 o'clocks, and that's the flower that I have in the corner. He had discovered that although leaf color had a dominant and a recessive gene, dominant being green and recessive being white, they didn't segregate according to the rules of Mendel's genetics, but instead they were dependent on the sex of the plant. In other words, if the pollen, which is male, was green, the leaf was green. If the pollen was white, the leaf was white. And this led to questions about how the sex of the parent led to inheritance. Correns' work had a distinct influence on Thomas Morgan and his study of fruit flies. We will be talking about Thomas Morgan and his work next. The year is 1910, and Thomas Morgan is working with fruit flies, specifically Drosophila melanogaster. Why did he choose fruit flies? First and foremost, they show sexual dimorphism. That means that males and females look different. Second, they only have 4 chromosomes, 3 autosomes and 1 sex chromosome, which makes them easy to work with. And third, they have a very short lifespan, only 10 days. This allows you to get lots and lots of generations in a very short amount of time. As Morgan was studying his fruit flies, he discovered that there was a white-eyed male. This is unique because the normal eye, or called wild eye, is red. And he wanted to know if this white-eyed trait could be inherited using Mendel's principle. So he began by crossing a mutant male, which is the white-eyed male, to a normal red-eyed female to see what would happen. This is a picture of the Punnett square from Morgan's work. This is the parent or P generation. The female information is on the side, showing her 2 red chromosomes. The male information is on the top, showing his 2 white chromosomes. And the smaller chromosome on the far right is showing that it's a Y. If you look in the boxes, you can see that all of the F1 generation, or all of the offspring, male and female, had red eyes. Morgan continued his studies by crossing a female and a male from the F1 generation. And you can see their information is heterozygous. This is the assumption because the females were both red chromosomed and the males were both white chromosomed. If you look at the F2 generation, only the males had white eyes. This led Morgan to wonder what had happened to the females. Was it possible that white-eyed females died? So this led him to his next part of the experiment, a test cross. A test cross is a cross between an individual that shows the dominant allele of a trait. That means that it's known to have at least one of the dominant genes. In this case, our female shows the dominant red gene because she has red eyes. That individual is crossed with another individual that is known to be homozygous recessive for the same trait. In other words, our male because he has white eyes, and we know that white eyes is a recessive trait. In this case, we took the female that we knew had one red gene, and we mated her to a male that we knew had two white genes. The result gave us males and females that had red eyes or white eyes. This proved that white-eyed females could live. It also told Morgan that eye color is linked to the X chromosome. That means that there is no color gene on the Y chromosome for any male. As you know, sex chromosomes are the chromosomes that are associated with an organism's determination of gender, being male or female. These sex chromosomes can vary according to species. Drosophila, or fruit flies, are like us. It takes two X chromosomes to be female, or one X and one Y to be male. An opposing example would be honeybees. One X makes an organism female, two X's make that organism male. And we talked about this when we talked about asexual reproduction and the Komodo dragons. A sex-linked trait, then, is a trait that is determined by a gene which is found on a sex chromosome. Most sex-linked traits are found on the X chromosome just because of size. Most Y chromosomes are very small, and so therefore they don't have a matching gene. So in the case of our fruit flies, the X chromosome had the color for white, but the Y chromosome didn't have a match, a dominant match to oppose it, which is why males showed up with white eyes more often than females. As you know, humans have 22 pairs of autosomes and one pair of sex chromosomes. The picture in the top left is a karyotype, and it's a karyotype of a female. The picture at the bottom is an electron microscope picture of a Y chromosome on the left and an X chromosome on the right. You can see the significant size difference between the two, and that's because the X chromosome provides the template for all of the body's necessary information. A human male Y chromosome actually only has 78 active genes, and very few of those genes are actually expressed. This is because they usually have a counterpart on the X chromosome that covers them up. One of the genes that is on the Y chromosome that isn't covered up is the gene for hairy ears. This is the gene that allows older men to grow hair from their ears. Another Y-related gene is the SRY gene. This is the gene that gives a man his maleness, the Adam's apple, the facial hair, the deeper voice. If part of the Y chromosome breaks off, and that part contains the SRY gene, and that part then attaches to an X chromosome in a female, this would happen during meiosis, that female will actually develop as a male even though she has two X chromosomes. If there is a mutation in an SRY gene, even though it remains attached to a Y chromosome, that male, even though he has that Y chromosome, will develop as a female. This shows you how vital the SRY gene is to the correct development of the human male. Next, I want to talk about the idea of dosage compensation. We've talked quite a bit in the previous chapters about the importance of having an equal number of chromosomes in the cells so you don't have too much information. It would seem then that since a female has 2 X chromosomes and a male only has 1, that she should have conflicting information, meaning that she would create twice as many proteins as a male, which would cause all sorts of genetic issues. But this doesn't happen. And the question is why? And the answer is this idea of dosage compensation. Dosage compensation is the cell's way of ensuring an equal expression of genes from the sex chromosomes, even though females have 2 X's and males have only 1. Early in the development of the female embryo, 1 X chromosome in each cell becomes condensed. When it becomes condensed, the genes are hidden, and that means that they cannot be used for protein synthesis. A condensed chromosome is inactive. We talked about that during the mitosis and meiosis chapters. A condensed chromosome, condensed during dosage compensation, is called a bar body. Which chromosome of the 2 X's is condensed and turned into a bar body is completely random. If a female is homozygous dominant or homozygous recessive for a particular trait, and one of those chromosomes becomes a bar body, the trait that is expressed isn't changed. So for example, if she's got dominant brown hair gene on either chromosome, it doesn't matter which one is turned off, she will still have dominant brown hair. If, however, she's heterozygous, which means she's got a dominant brown gene on one chromosome and a recessive blonde gene on the other, it will make a distinct difference which trait is expressed depending on which X has been randomly inactivated. Calico cats are a classic example of this idea of dosage compensation. They also show the idea of epistasis. I will talk about both of those in class. The chromosomal theory of inheritance says that the majority of the information that we inherit from our parents comes from the DNA, which is found in the nucleus. We can, however, get certain genetic information from other DNA. That's the DNA that's found in the mitochondria. Remember, we talked about the endosymbiotic theory way at the beginning of the year and how mitochondria and chloroplasts have their own DNA. When a zygote is formed, remember that's egg plus sperm, each parent gives an equal contribution of genetic material. But as you know, the majority of that genetic information, particularly information about the creation of organelles, comes from the mother. Because of that, the information about mitochondria contains maternal DNA, or information from your mother. So you get information from the DNA of your nucleus, which is a combination of mom and dad, and you get additional information from your mom through your mom's mitochondrial DNA. Mitochondrial DNA is not as specific as the nucleus DNA. Nucleus DNA can specifically tell you this individual is my father, this individual is my mother. DNA in the mitochondria can only tell you if a woman is from your family line. It can't even tell you sister or aunt or grandmother. It can just tell you very basic relationships. Genomic imprinting is the autosomal version of a bar body. A chromosome has been inactivated, usually through the epigenetic process we discussed in Chapter 12. These genes that have been inactivated are called imprinted genes. When that particular gene has become inactivated, the organism is essentially haploid for that gene because it has only one working version. What's interesting about genomic imprinting is that the same gene turned off can produce a different result depending upon whether it was turned off in the male version of the chromosome or in the female version of the chromosome. A human example involves chromosome 15. In this case, if Dad's chromosome number 15 has experienced a partial deletion and Mom's chromosome number 15 has been imprinted or inactivated, the child will have Prader-Willi syndrome. Prader, P-R-A-D-E-R, Willi, W-I-L-L-I. This will make the child short, overweight, have difficulty breathing, slight mental retardation, and obsessive-compulsive disorder. If Mom has the deletion of chromosome 15, or partial deletion, and Dad's chromosome has been imprinted, the child will have Angelman syndrome, A-N-G-E-L-M-A-N. In this case, their entire physical development will be delayed. They will have severe mental retardation, hyperactivity, and jerky body movement. Sometimes the loss of imprinting can cause problems. In other words, there are some genes that are supposed to be inactivated. If that inactivation is lost, so an inactivation is inactivated, making a gene actually work, you can also have problems. So in other words, a normally silent gene begins to work. This could cause developmental problems, growth issues, or cancer. This is a review of nondisjunction. Remember, nondisjunction is the failure of chromosomes or chromatid to separate properly. That means that during the synapsis portion, anaphase 1, they didn't separate from pairs to single chromosomes, or during anaphase 2, the sister chromatid did not separate. An aneuploid gamete is a gamete that has the wrong number of chromosomes. Remember, monosomy means you're missing one, trisomy means you have an extra. Nondisjunction can have a wide range of effects on the organism. The effects can be mild, they can be severe, or they can cause spontaneous death. Data suggests that 35% of human miscarriages occur due to some form of aneuploidy. This is a karyotype of a male with trisomy 21 or Down syndrome. It occurred either through a translocation of a portion of chromosome 21 or the complete nondisjunction of chromosome 21. You already know what the traits are from a previous chapter, so I'm going to move on to Edwards syndrome, which is trisomy 18. Trisomy 18, or Edwards syndrome, occurs in about 1 out of every 3,000 live births. 50% of babies will be born stillborn, with baby boys having a higher stillborn rate than baby girls. Less than 10% of children with Edwards syndrome survive to their first birthday. A small number of adults, usually girls, will live up to as much as 30, but they must have extensive caregiving. Some of the symptoms include heart defects, kidney defects, small heads, severe developmental delays, and an esophagus that doesn't connect to the stomach. These individuals require a great deal of hospitalization to help them to make it even to 20 or 30. Nondisjunction of the sex chromosomes can also occur. The result of nondisjunction of sex chromosomes is both similar and different to nondisjunction of the autosomes. It's similar in that both forms of nondisjunction can affect the physical body, such as height and weight, but nondisjunction of the sex chromosomes, because of the nature of what the chromosomes involve, can also affect the sexual organs and other traits that are related to gender. This is the end of Chapter 13, Sexual Inheritance.